Okay, good afternoon, everybody, and welcome to the Statistical Mechanics in Chemistry and Biology SMCB seminar series. So we have been running this seminar series under the mentorship of Professor Bhiman Bagchi. I'm sure all of you know it by now. And there are three coordinators, myself, and then Professor Ranjit Bishwas from SN Bose Center, and Dr. Rajiv Bishwas from IIT Tirupati. So we have been running this over 3 years now. There have been more than 30 lectures. The goal is to communicate to the young students and postdocs and maybe even young researchers and talk about the cutting edge applications of statistical mechanics to the broad area of chemistry and biology. And it's a pleasure to have today young speakers, one young faculty and another postdoctoral researcher. The faculty is Dr. Debayan Chakraborty from Math Science, Chennai, and postdoctoral researcher Dr. Sayantan Mondal from Harvard University. Now, before we start the scientific presentations, I request our mentor, Professor Bhiman Bagchi, to say a few words about some of the future plans of this SMCB platform. So I request Professor Bagchi to say.
Thank you, Sumon, and thank you, Ranjit and Rajiv, for running this quite smoothly and for pretty long time now. And it is actually, when it was done, seminar series was one of the, now it is, the number is already 46. I think we'll probably cross 50 very soon. Okay, anyway, but the idea was not just seminar series. Seminar series is one of the activities. It is basically to have a society. I always love to talk with some anecdotes, some stories. So here this story comes from my good young friend, Proval Maiti, and a very new one. I was talking to him last week, and he said that there was some award. I won't go into the detail because I don't want to very particular things. There was an award somewhere, an international award. He was a member of the committee, and there was this one of our SMCB member was shortlisted, and but then ultimately, I think didn't make it to last 4 or so, 8 or whatever. The reason being that there is a huge number of papers being published from few universities, which are just overwhelming. And they border on essentially physical materials chemistry. Materials chemistry had this advantage, they can come like chemistry. They can be physical chemist, inorganic chemist, or organic chemist. Now, so that is a serious concern. Then I was talking with another physicist, condensed matter, quantum, who said that now even the very top guys, and one name is whom I have followed for a long time, is Subit Sachdev. And he did PhD at Harvard with somebody I forgot. Oh, yes, David Nelson. And then another boy, Ocean, is somebody who moved to Harvard from Berkeley. Both of them published about 30 papers. And now in quantum condensed matter, this is not a small thing, right? And they are not supposed to publish that many papers. But their logic, I heard, is that in turn, that there is so much competition. So it is that is something we are going through now, and we have to live with that. Now, one of the things that they do everywhere is they do a lot of collaboration, and many times faculty work among themselves, and if faculty work among themselves, relatively things can be done relatively fast, even with 1 or 2 students' involvement. So as I was saying, when this SMCB came into being or was kind of formalized, then it does not just a seminar series. It's essentially a society for very like-minded people, and one of the things that we always thought that we want, without wasting any more time, because I don't want to take the valuable time, is to have a preprint library. We had that in TCS. In the very beginning, one of the first thing we did in TCS was the preprint library. And first two were run by Professor Sattamurthy and myself. First Professor Sattamurthy, then myself. Then it was given to somebody and it died. But it was very successful. And those days we used to get the preprints and then we used to write the abstract, or I told them to send the abstract by email. So what I'm saying that the next step we should certainly try and think of starting a preprint library. The modalities, of course, we have to have a web page where we have to have a link, and I think that things can be taken care of. And one of the young people should take care of that. Because see, one of the things is that in your own field, from your own country, if you get and you see that you can do some work and collaboration, that would really help you. And to create not only a society of yourself, certain interaction and certain visibility. Otherwise, you will be just we should not become too small a community. And some activity is required, not just seminar series, but also preprint library. We are going to do an in-person meeting, thanks to Rajiv Biswas and Devashish Mondal in Tirupati with this December, and that I think they have announced already. So certain amount of this kind of activity is very important to make it a viable community. You know, I don't know how long more I'll remain involved in these things, but or will be able to get involved in these things. But I think this must be stable, and certain amount of activity is required to keep it stable. Like you need to in an orbit, to be stay in an orbit, you must move in a certain velocity. So activity really helps. And preprint library is one of the best things we had when you were TCS. And modalities can be done. Somebody has to send the preprints, not the preprints as the abstract, or maybe an extended abstract. It will be very nice to have an extended abstract. But then everybody get to know what is happening, because anyway, you don't read too many journals anymore after it came online. But this can help. That's all I had to say. And I just don't want to take in time. You know, as you grow older and older, you start talking a bit more and more. But this is an idea I wanted to, whose time really has come. And that is with that preprint library. I give it back to Suman, and I see there's already 54. That's a very good number. Okay, Suman.
Thank you, Professor Bagchi. And that is an excellent idea to have a preprint library. We'll have to figure out the logistics of it very soon. So let's now start the academic discussion. So our first speaker today is rising young star and fresh to Indian academia, Dr. Debayan Chakraborty. So let me briefly introduce him. So Debayan did his master's from Oxford, University of Oxford in theoretical chemistry. Then he did his PhD from University of Cambridge under the supervision of Professor David Wells. So there he explored this area of energy landscape framework to understand protein folding and RNA folding and so on. Then he did his postdoc with Professor Dave Tirumalai at University of Texas, Austin. There he developed coarse-grained models to look at protein DNA interactions, IDPs, amyloid integration, so large, diverse range of problems. He came back to India almost a year back. He's now faculty in the theoretical physics unit of Institute of Mathematical Sciences in Chennai. And again, he is working on a wide range of problems at the interface of physics, biology, and chemistry. In particular, he's interested in protein folding, allostery, amyloid aggregation, and chromatin organization. So today, he's going to give us some coffee, a coarse-grained force field for simulations of DNA protein complexes. Debayan, all yours.
Yeah,
so
thank you.
Yeah, you have 40 minutes. I'll give you a warning, maybe 5 minutes in early.
So, sure. Okay. Thank you so much, Shumanda, for the kind introduction and for inviting me. And I'm also very grateful to Professor Bagchi and the other mentors who have put together this amazing, forum where young faculty like me and also postdoc scholars can sort of present their research work and get some critical feedback. So today I'm going to talk about coffee, one of our recent works, which sort of describes the coarse-grained force field that we have developed to simulate DNA protein complexes. And our purpose of developing coffee was to have a generalized framework for biomolecular simulations. So let me first acknowledge my postdoctoral mentor, Professor Dave Thurumalai at University of Texas, Austin. And let me also acknowledge the contribution of Dr. Bolaka Mondal, who is a postdoc currently at University of Texas, Austin. And she was the one who actually helped us to speed up some of the simulations by implementing this code into GPU-enabled softwares like OpenML. Okay, so without any further ado, let me just start. And just to point out, the work that I'm going to present today has recently been published in JCTC last couple of months back. So when we are thinking about coffee, a nice cup of coffee, the question that I ask is what actually makes a nice cup of coffee? And the answer could be a judicious combination of ingredients. It's very important. You need to have really nice coffee beans. You need to have, for those who want to have milk in their coffee, you need to have good quality milk. And of course, who like sugar in their coffee, you need to have good quality of sugar. And essentially, when we are thinking of a nice cup of coffee, we want to go to the nearest Starbucks or CCD or any other place you can think of. But the idea today is not to advertise these places. So for the purpose of this talk, coffee will be an acronym for this, coarse-grained force field for energy estimation. But the key point here is even for this coffee, the ingredients and their combination is extremely important. Now you may ask, why did we actually choose the name coffee for our force field? And the simple answer would be we were actually just trying to keep the tradition alive. I mean, there has been a long tradition in the area of biomolecular simulations, particularly to name force fields after certain kinds of drinks. For example, many of you might have heard about the Martini force field. Then there is the absinthe model, which has been developed by Rohit Papu and co-workers, and more recently, the Lassi model for simulating phase separation in multivalent proteins. Now, in our cup of coffee, what we have done is we have adopted a coarse graining route. Now, when you have to do some biomolecular simulation, let's say it could be a protein, it could be a DNA molecule, or it could be a complex of a protein and a DNA molecule. So you are always tempted to do something like all atom molecular dynamic simulations, where you sort of describe each of the interactions at the atomistic level. You have some description of the solvent that is solvating the protein molecule as well as the ions. So as you can see here, everything is represented in the atomistic resolution. But on the other hand, there are some problems associated with these kinds of representations, at least with the type of computing power that we currently have. You cannot really simulate very large systems over very long time scales. So what is one of the alternatives? One of the alternatives is actually to go over to a much simpler model, or sort of coarse grain over unnecessary degrees of freedom, which are not suitable to the problem of interest. So this gives what is called, is there a question in the chat? I see.
No, it's okay. I just ask people to post their questions.
Okay, fine. Okay. So yeah, so one of the alternatives would be a coarse grain model where basically you have a much simplified representation of the system. You throw away unnecessary degrees of freedom. And what that does is, because there is a huge reduction in the degrees of freedom, you can actually go over to much larger length scales and longer time scales. So in terms of the underlying energy landscape, what does it do? So let's say this is my all-atom energy landscape. As you can see, it has a lot of this minima. It's very rugged with a lot of features. However, when you do a coarse graining, when you are sort of thinking about a much simplified model, what it does is basically makes this energy landscape somewhat less bumpy. So you have sort of removed many of the undulations that you see on its energy landscape. But the key thing for any robust coarse graining approach should be it should still be able to sort of capture the underlying physics of the system. So it should still retain some of the basic features of this landscape. So as you can see in this mapping, some of the key minima that you see here are basically retained. So this is one of the purposes of coarse graining, that although you are simplifying the system a lot, you still hope to retain the underlying physics. Now, in the coffee that we are brewing, what we are essentially doing is we are combining two things. One is what is called a coarse grain model of DNA, where you see it's a very simplified representation of the DNA double helix in terms of some spherical beads. And with that, we combine a coarse grain model of a protein. where again, instead of representing all the atoms of the amino acids, we are simply representing the amino acids by certain number of beads. So let me describe these two ingredients which are important for brewing our coffee in somewhat more detail. So this is the ingredient one, which is there are three interaction site model for DNA. So what happens is, let me just show you a comparison of the all atom representation of DNA, which is shown on the left hand side, and the three interaction site DNA representation. So what we are essentially doing is we are representing every nucleotide by three interaction sites or three beads. So one of the beads is centered on the phosphate group, one is centered on the sugar group, and the other one is centered on the nucleobase. So this is the coarse-graining scheme that we have used. And to describe the force field, we have different components that are there. As you can see, there is a bonded term, which is simply a harmonic potential, which describes how the different atoms, or in this case, the beads, are connected to one another. There is the term describing the angular interactions, which is again harmonic. Then there is an excluded volume term, which you actually need to ensure that your different beads do not come and sit on top of each other and lead to unphysical interactions. And the key things here are these last three terms, which are the electrostatic interactions, which describe how your different phosphate beads interact with one another. There's a term from the stacking interactions. So as you know, in DNA, different nucleobases are stacked on top of one another. So this term describes the stacking interactions among the different beads. And of course, the key thing here is the hydrogen bonding interaction. So as we know, in DNA, you have Watson-Crick base pairing mostly. So between A, T, and GC. So this is the hydrogen bonding term, which describes those sorts of interactions. And important here is the stacking and the hydrogen bonding, as well as the bonded and angular terms, are sequence dependent. So depending on what DNA sequence you deal with, these different energy terms will be different. Okay. Now, we have recently sort of described this model a couple of years back, so it appeared in this Journal of Chemical Theory and Computation. So we have shown that this DNA model actually recapitulates many of the experimental properties that are well established for both single-stranded DNA and double-stranded DNA. So here I show you some of the representative results. So here we have compared what is called the effective scaling exponent nu. So the way you extract the scaling exponent is you have a plot of radius of gyration versus the size of the polymer, and you can fit it to the Flory scaling law to extract these exponents. And this is what typically was done by experiments of Pollack and co-workers. So these circles are basically the Flory exponents that were sort of extracted from the results obtained from Sachs experiments. And the blue solid circles that you see are from artist DNA simulations. So as you can see, it sort of gets you very close to the experimental estimates. Now, if you have a completely random coil behavior, the Flory scaling exponent would be somewhere close to about 0.6 or 0.588. So interestingly, you see, like, although you keep on increasing the salt concentration, at no salt concentration, the poly DA chain really behaves like a random coil, but at some salt concentration for poly DT, you see that it crosses this dashed line. So this again hints at the sequence specific behavior that our TIS DNA model is capturing. The other one of the other things we looked at is basically the melting of DNA hairpins. So the melting temperature for DNA hairpins of different sequences were sort of obtained from experiments. And we tried to see how well our TIS DNA model is doing. And as you can see from this table here, it gets rather close for most of the sequences for which the melting temperatures are known from experiments. Now, the ingredient 2 of our force field is basically this coarse-grained model for proteins. So this is called the self-organized polymer model, and it was actually developed previously by Dev Thirumalai's group, and Govardhan Reddy, who is in IISc. He has worked also extensively on this model. They have developed different variants of this model. So let me describe the variant that we are using. So here, basically, each of these amino acids are represented by two beads. So as you can see, one bead is actually on the backbone, the C alpha atom, and the other bead is centered on the center of mass of the side chain. So the reason for showing these two side chains in different colors will become very clear, right, in a few minutes. So this is the force field that we have for the self-organizing polymer model with side chains. So again, you have a Finney potential, which describes the chain connectivity between the different amino acid beads. you have a repulsion term. Again, the purpose of this repulsion is to ensure that you do not have two beads sitting on top of one another and creating unphysical overlaps. The key interactions in this model are the side chain, C alpha, and C alpha side chain interactions. And you also have a term coming from the electrostatic interactions between the different side chain beads, again, which is described according to the Debye-Huckel theory. Now, the key part to note here is this: these three interaction terms that I have here, this side chain, C alpha, and C alpha side chain, they are in the form of these Lennard-Jones potentials, and they have different energy scales. And basically, the energy scales were optimized in this work by Reddy and Thirumalai, and we have just used those values in our case. And the important part to note here is this side chain interactions. You have this epsilon I, which is called often the Bittencourt-Thirumalai matrix, and it's like kind of like a statistical potential, where you can derive the different contact energies based on the frequency of contacts among different amino acid residues. So essentially, what it means is this epsilon I will be different for different amino acid side chain beads, and that basically encodes the sequence specificity of this self-organized polymer model. And as I just said, we have basically used this epsilon values from this paper by Govardhan Reddy and Dale. Now, this SOPSC model has been used rather successfully, actually, to look at a lot of different problems in protein folding using the folding of ubiquitin at different pH conditions. And they also showed the folding of this green fluorescent protein, where one could find out different routes to the native state. Now, when coming back to coffee, this is what we have then. We basically have the force field for DNA. We have a force field for the protein, which was developed earlier by Reddy and Thirumalai. And now what we really need to add is some way of describing the DNA-protein interactions. So this basically gives you the overall force field. Now, how do we describe the DNA-protein interactions? So naively, one might think that one can split it up into two components. One is an electrostatic component, which simply describes how your phosphate beads are going to interact with the different charged amino acids, like, for example, arginine, aspartic acid, glutamic acid, and so on. And this is the non-electrostatic component of the DNA-protein interactions, which is simply the van der Waals interactions. Now, how do we describe this non-electrostatic component then? So one can write it down in this way, where you have a component arising from the native-like interactions, and you then have another potential term corresponding to the non-native interactions. So here, by native interactions, I mean the interactions which are already present in your crystal structure. So you can sort of define the native interactions in terms of the residues that are already in contact in the crystal structure. So typically, it's like a Go model with a certain bias towards the experimentally known crystal structure or the NMR structure. And this lambda DNA pro basically modulates the strength of this Go potential or the contact potential. Now, the native part of this interaction, it can be written in this way, again in the form of a Lennard-Jones potential. And the key term is this epsilon xi. So these are the energy scales for this native-like interactions. And so the question is, how can one determine these energy scales? So one way of determining these energy scales is again from the contact statistics from a database of DNA protein structures. So for that, what we did was we considered a non-redundant data set of DNA protein complexes. So the idea behind using a non-redundant data set is you do not want to have any kind of bias in your statistics. So in non-redundant data set, basically you have DNA protein complexes for which there is very, very less sequence similarity in going from one structure to another. So that sort of ensures that the bias is minimized. So essentially, when you have this PDB database, you can actually count the number of times different amino acid and nucleotides are in contact with one another. And once you do that counting exercise, you can actually simply find this free energy of contact formation between nucleobus X and the amino acid Y in this way. So this is how it is described. So, and one key thing here is when you are defining a statistical potential of in this way is what reference do you choose? So the easiest way to think about a reference state is what would be the probability that my amino acid of type X would be in contact with nucleobase type Y if they were to occur in a random fashion. Okay, so and this is just the product of the individual probability. So this is how you define the free energy of contact between X and Y, and you can relate these energy scales to this free energy of contact formation. Then the idea is you also have this lambda parameter in your force field. So how does one choose an appropriate value of lambda for the force field? Because that ultimately tunes the strength of the native interactions in our case. So what we did was we basically chose a very high resolution structure of this human TBP core domain, which is complex with DNA. This is a rather small molecule, and we wanted to see like for what value of lambda are we actually optimizing the root mean square error between the experimental B factors and the simulated B factors. So the B factors effectively tell you about the fluctuations at the residue level, and we wanted to see like how to choose lambda so that we have a good agreement with what is reported in the X-ray structure. And it turns out that 0.15 is a good enough number in our case, and we chose that as we chose that value of lambda, and we keep it fixed for the rest of the complexes and the rest of the sequences. Once it is optimized, we do not tweak it any further. Now we wanted to see, after we sort of choose an optimal value of lambda based on this structure, based on this high resolution crystal structure, how does it do for other DNA protein complexes? Is there any property, experimental property, that it is predicting in quantitative agreement or not? So for that purpose, what we did was we looked at these 3 different DNA protein complexes, and we computed what is called a scattering curve from simulations, and we tried to compare that with experiments. Now, Sachs curves actually tell you about the overall size of the DNA protein complex, which in this case is the radius of gyration. So we can compute the radius of gyration from our simulations, and we found that it is actually in very good agreement with what was predicted from the Sachs experiments in all 3 cases. And as you can see, these DNA protein complexes are of different lengths. In one case, it is 209 residues, and the other 2 are much longer. Now, one might argue that, well, with Sachs, you are getting the overall dimensions or the overall size of your DNA protein correct. But how about the local residual structure or the local residue level information? How does it do for those kind of scenarios or experiments? So one way of actually testing that is to compare it with respect to chemical shifts. So we found some chemical shift data for this lap depressor complex with operator DNA, and we sort of looked at the chemical shifts that we measured from the simulation with those reported from experiments. And again, as you can see, for both the subunits that are shown in orange and magenta, the ones obtained from simulations are in very, very good agreement with what you see from experiments. And here again, here I show you a comparison of the contact maps that we get from the NMR ensemble and what the ensemble that we get from simulations. Again, you see that most of the features we see are pretty much equivalent to what you see in case of the NMR. So let me just tell you like what was the motivation of actually developing this force field coffee. So one of the things we were very interested in understanding is genome organization. So as you know, in our cells, we have about 2 meters of DNA, which is sort of compressed into within a very tiny volume of the nucleus. So the question is, how does it happen? So what people know is the organization is kind of hierarchical, and it happens at different stages. So the first level of the organization is where you have about 147 base pairs of this double-stranded DNA, which is wrapped around this core of histone proteins. So this is an octameric core where you have 2 copies of each of these different histones, which are shown here in different colors. So these are all mostly helices. And in addition to that, you also have what are called histone tails. So these are all intrinsically disordered proteins. And these histone tails actually mediate interactions between different, as you see, nucleosome units in the overall chromatin fiber. So what you have is your double-stranded DNA first wrapped around to form this DNA protein complex called the nucleosome, and then you have this nucleosomes kind of threaded together and further folded to form the chromatin, and then you go up to this level. So we wanted to understand basically the organization at this level, which is the nucleosome. Now, what is known is from experiments and a lot of previous work that nucleosomes are very dynamic. So essentially what I show here is a wrapped configuration of the nucleosome, but transiently what happens is some of the DNA can sort of come out, and when this happens, it is sort of it can be read by the transcription machinery. So it is, in a way, you can say that this comes out on and off based on the requirements. So we wanted to understand typically this transition, how this wrapped nucleosome basically unwraps. And the motivation also comes from some of the recent in vitro experiments of Polak and co-workers. So they, in fact, what they did was they induced this nucleosome unwrapping or disassembly by sort of systematically increasing the salt concentration. So naively, what we would expect is, in this kind of complex, it is stabilized by electrostatic interactions between the different charged histone proteins and the phosphate groups. So when you are adding more and more salt, these electrostatic interactions actually become somewhat weaker. So when you have low salt concentration, you have this nucleosome core particle which is completely wrapped. But as you are increasing the salt concentration, you sort of induce more and more breathing motions of the DNA. And finally, at certain salt concentrations, you start losing some of the histone proteins, and even the DNA sort of comes out from the core structure. So this was one of the motivations for our study as well. And interestingly, a few years ago, there were some cryo-EM structures of this partially unwrapped nucleosomes which were published at high salt concentrations. And what they showed was typically the nucleosome kind of unwraps from one of the ends and not both ends. So this is what I will touch upon again in the next slide. So this brings me back to some very seminal experiments by Bustamante and co-workers on nucleosomes. So what they showed was you can systematically unwrap a nucleosome as a function of force. So if you are in this low force regime, what happens is the outer turn of the nucleosome unwraps. But as you keep increasing the force, you will hit a point where the inner turn also starts unwrapping. So the low force regime is somewhat around 5 piconewtons, but once you start pulling it beyond 8 piconewtons, you also see the inner turn of the nucleosome unwrapping. But in their experiments, they did not really talk about which end of the nucleosome is coming out. And this was actually discussed in these, again, very seminal experiments of T.J. Ha and co-workers. In fact, what they showed is the unwrapping that you see in nucleosome is somewhat asymmetric. In other words, if you have, let's say, the local DNA sequence is different on both ends of the nucleosome, the more rigid sequence will actually unwrap first before the more flexible end. Because if you have a rigid sequence, it is not able to actually accommodate the sharp bending or the turn that you see in the nucleosome structure. So this was a very interesting work of T.J. Ha and co-workers. And more recently, there was some very nice work again from Polak's group, where in fact they did some SAX experiments at different salt concentrations, and then they tried to fit different pools of nucleosome conformations to those SAX data, and they've sort of predicted or opined that the unwrapping can happen through multiple different pathways. You could have a unwrapping pathway involving this kind of an intermediate, which is a symmetric intermediate where both the ends of DNA, same amount of DNA unwraps from both the ends, or you could also have what is this asymmetric unwrapping, where typically you have the unwrapping happening from one of the ends of the nucleosome. So we wanted to understand this process in somewhat more detail, and of course, as you will know, there's some finer details of this nucleosome dynamics, especially the unwrapping dynamics. It would be very, very hard for many experiments because of the limited spatial and temporal resolution. And this is where actually computer simulations can start playing a huge role. In fact, there is some very nice work using atomistic simulations of nucleosome. Let me point out, they are very, very challenging to do, even with all these advances in computer hardware, as I was mentioning in one of my introductory slides. In fact, for both these studies that I shown here, the specialized computer hardware that is only available in this Anton 2 supercomputer maintained by DE Shaw group was used. So here, this Aximintiv and co-workers basically, again, with atomistic simulations, they showed that, the nucleosome tends to unwrap from only one of the ends. But as you would see, they had to use very, very high salt concentrations to actually make unwrapping happen on the simulation timescale. On the other hand, recently in 2021, there was another very interesting study which did about 15 microsecond of molecular dynamic simulations. And this was at about 150 millimolar salt concentrations. And then again, they were able to sort of resolve many of the different DNA breathing motions and unwrapping events. So of course, I mean, we did not have access to Anton tools, and we had to sort of resort more minimalist coarse-grained models to get anywhere. And as I say here, even 15 microseconds may actually not be enough to see the full unwrapping process. So the first thing we try to see is like how well does our coffee force field do for nucleosomes. So we chose this this nucleosome complex for which the high resolution structure was available from the PDB database. So here basically you have the DNA sequences, this alpha satellite DNA sequence, and you have an octamidic histone core. And interestingly, in this X-ray structure, they also were able to resolve the different histone tails. So the first thing we wanted to see is if we compare the simulated scattering curve with the one obtained from experiments, how well we are doing. And we find that, as you can see, the Sachs curve is pretty well reproduced. And even the radius of gyration that we estimate from the simulations is very, very close to what was estimated from experiments. But this is just to do with the fully wrapped nucleosome. The other thing we wanted to find out is what happens when we actually increase the salt concentration in our simulations. Are we able to capture anything to do with salt-induced unwrapping that was suggested in the experiments of Polak and co-workers? And this is what we did. We basically simulated the nucleosome at different salt concentrations from 0.1 molar to about 1 molar. So as you can see here, the probability distributions are in the form of the number of unwrapped base pairs. So if you are at very low salt concentration, you see that the nucleosome is fully wrapped. I mean, it's sharply peaked around 0, but as you keep increasing the salt concentrations, you start populating different partially unwrapped states. And interestingly, what we found is we found populations of both asymmetrically unwrapped states where you see the nucleosome kind of unwrapping only from one end, or symmetrically unwrapped states where you see like both of the ends contributing. So basically, we did not yet kind of do a very systematic quantification in terms of if there are any switches in the populations of asymmetric or symmetrically unwrapped states as you sort of start changing the salt concentration, but that is a work in progress. And then we also try to understand what happens. So as you can see here, you have the histone tails that are emanating from the nucleosome core. So the question we asked ourselves is, what happens if we chop off those histone tails? So interestingly, what happens is, if we again simulated the nucleosomes at the same range of salt concentration, starting from 0.1 molar to 1 molar, and we see that once you chop off the histone tails, the fully wrapped nucleosome is no longer stable, even at low salt concentrations. As you see from this probability distribution, you will always have some amount of unwrapping going on. And then as you keep increasing the salt concentration, you again start populating different kind of partially unwrapped states. And interestingly, when you are at very high salt concentration between 0.8 to 1 molar, you can fully disassemble the nucleosome. So as you can see here, the histone core basically comes off from the DNA. So, and here I show basically the average number of unwrapped base pairs as a function of salt concentration for both the scenarios when we have the tails and when we don't have the tails. So as you can see, when we have the tails, the average number is always much smaller as opposed to the scenario when you don't have the tails. Okay. So this now brings me to the asymmetry of the unwrapping. As I said that there are these different experiments from TJ Haas group and even the salt-induced disassembly of nucleosomes, which was sort of studied by Polak and co-workers using Sachs experiments. So they kind of hinted at some kind of asymmetry in the unwrap. So we wanted to understand this asymmetry in some more detail. So what we did was we basically defined what is called the asymmetry parameter, which is nothing but the ratio of the number of base pairs that is unwrapping from one side divided by the total number of base pairs that is unwrapped. Okay, and we looked at the free energy free energy landscape at about 0.2 molar. So here you see in this salt concentration, there is an equal probability of kind of populating the both the asymmetric and the symmetrically unwrapped states, as you can see here. Now, when we increase the salt concentration somewhat to about 0.5 molar, you see that there is one side which is kind of trying to win this time. So you have a right dominated side which is winning at 0.5 molar. So what we believe is happening is once a substantial amount of DNA has unwrapped from one of the ends, somehow there is a communication to the other side, and then there is a rearrangement which sort of tries to hold the DNA and protein much more tightly at the other end. So that prevents the other end from unwrapping. So let me tell you, we haven't really fully understood the details of it, but this is what we think is happening. Now, the other thing we wanted to see is what happens if we chop off the histone tails. Is there any loss of asymmetry? And this is what we see. Like once we remove the histone tails, the asymmetry basically becomes somewhat weaker, which again hints at the role of this histone tail DNA interactions in sort of controlling the asymmetry of the unwrapping process. And the other aspect we looked at, which is somewhat interesting, is what happens if we do some mutations at the key locations, or these are called the superhelical locations, where actually the histone proteins kind of touch the DNA groove. So we wanted to understand what happens if we do some mutations at these locations. So here, basically, the contacts that you see in these green, denoted by these green spheres, are all arginine phosphate salt bridges. And I mean, people have pointed out that these are actually very important for the stability of the nucleosomes. And in fact, if you disrupt these contacts, these kind of mutations people have seen in yeast, and it is known to increase the flexibility of the nucleosome structure. So we wanted to see if our model kind of captures that. So what we did is we mutated the arginines to lysines at all these superhelical locations. So effectively what it does, it sort of doesn't impact the electrostatic interactions, but changes the chemical nature of the contact. And in fact, what we see is, I show you the probability distribution at 0.5 molar. So for the canonical nucleosome, you see where you have the arginine phosphate interaction, it's a bimodal distribution where you have one peak corresponding to about 20 unwrapped base pairs and another one corresponding to 60. But once you do the mutations at these locations from arginine to lysine, you see a shift in the population. And you see, although it's still a bimodal distribution, now there's a switch in the populations, and you have more number of bases which are sort of unwrapping, and you see the major peak at around 80. And let me point out that this is a purely chemical effect which is basically arising, or which we are able to capture because we have used this sequence specific statistical potential, sequence specific contact energies for the different DNA protein contacts. And this is something which is actually missing in most of the coarse-grained models of the resolution that we use. So you do not, I mean, those models, they basically assume the same energy scale for all the different interactions. So this is a very key ingredient, or you can say a unique ingredient of the model that we have developed. And also we have done some work. So you might argue that right now we are actually describing the electrostatics of the salt effects mostly using this Debye Huckel, but it's not really a very good approximation when you go to intermediate to high salt concentrations. So is there a way to sort of correct that? So what now we are doing is we have actually developed a model where we include these ions explicitly in the simulation. So we have sort of we have developed this 3 interaction site DNA ion model. So now we just have the DNA model with the ions, as you can see here. So it can work with both monovalent and divalent cations. And here we show I show you some of the recent results. Here, the radius of gyration for this DT30 sequence as a function of the salt concentration. And in the inset, I show how it agrees with, again, experiments from Pollack and co-workers. So you see, it not only distinguishes between magnesium and calcium, but it also distinguishes again between DT30 and DA30. And Other thing I wanted to show is, what happens if we go to double-stranded DNA? So we looked at this 25 base pair double-stranded DNA sequence for which some SATS data was available at two different salt concentrations, 3 millimolar magnesium and 16 millimolar magnesium. And here again, as you see, the simulated SATS curve sort of agrees quite well with the experiments. So the idea is now to actually include these ions explicitly within this force field that we have for the DNA protein complexes. Particularly, we are very interested to look at the effect of divalent cations like magnesium and calcium, how it affects the unwrapping of nucleosome, and whether again it has a profound effect on the asymmetry. So this is a work in progress. And let me give a summary of whatever I have spoken about. So what I told you is we have sort of brewed this force field, which we christened coffee, by combining two robust coarse-grained potentials for DNA and protein. So these were already published independently. So we just try to combine them in a way that it sort of becomes consistent with, it sort of recapitulates many of the experimental data that is published for different complexes. And as I showed you, we were able to recapitulate some of the SAC spectra that people have reported from experiments, and also the salt-induced destabilization of nucleosomes, which many experiments from Pollack's groups and others have suggested. And interestingly, what we find is the salt concentration actually has some role to play on the direction of unwrapping and as well as the asymmetry. And in fact, the histone tails also seems to have a profound, the presence or the absence of histone tails also seems to have some effect on asymmetry. And I think another key point is our model is able to capture the effect of mutations. And this is, I would say, a unique feature given the resolution of our model. I mean, there are many other high resolution models which can have captured these effects, but for a model of our resolution, I think it's a unique feature. And with that, I would end, and I would be very happy to answer any questions. Thank you so much.
Thank you, Debayan, for an excellent talk. Now the floor is open for question. So I see several questions in the chat box. So maybe I'll start reading them, but if you'd like to ask them yourself, please raise your hand. So I'll start with a question from Konkona. So how do you represent water to capture nucleosome unwrapping process in CG simulation? Secondly, what was the time length of the simulation for the same?
Okay, this is a very good question again. We do not have any explicit representation of water. So we are actually doing Langevin dynamic simulations where you can very crudely kind of capture the viscous effects, but that is about it. So the idea is now it is well known that there can be a lot of interesting things going on regarding solvation as to do with nucleosome. So we want to actually integrate our model with some of these coarse-grained water models that the other groups have published. For example, the mono-water model that Molinero's group has published. So this is something we want to do. Thanks for this question. It's a very good point.
Thank you. Next question from Anushree. How the increase in ion concentration is resulting in the unwrapping? What could be the microscopic insight?
Okay, so what is happening is, as I said, that most of the interactions that you see, or the stability that you see in the nucleosome structure, you have this phosphate groups which are negatively charged. They are interacting with the histone proteins. Actually, the histone core has a net positive charge. Okay, so as you are increasing the salt concentration, the electrostatic, the interactions become weaker. So that sort of results in the unwrapping, nucleosome unwrapping. So you are weakening the electrostatic interactions between the histone proteins and the DNA.
Thank you. Next question was from Rajesh Murarka. I think that is kind of similar. Rajesh, do you have any additional question to add?
No, I think I've asked similar question.
Yeah, so this is also a very good question. So the arginine phosphate interactions, to actually disrupt it, you need a much higher salt concentration as opposed to lysine phosphate, because the lysine phosphate interaction scales are somewhat weaker.
So that difference is also reflected in that mutation.
Yes,
so
exactly.
And to the electrostatic interaction also.
Yes. So you have the Yeah. So here the charge is the same, but then you also have a non-electrostatic component as well.
Right. Yes.
The major component is still the electrostatics, but you cannot discount the non-electrostatic component. So here that effect you can see.
Shongita, there are many questions. Anyway, I'll start reading. So the first one is selected lambda DNA pro is generalized or it changes with the protein DNA complex?
Okay, so it is generalized. So as I said, once we fix it by sort of looking at the root mean square error of this small crystal structure that we used. So the idea was that, like, once we sort of optimize it for one crystal structure, let us keep it the same and not tweak it and see how well we do. And okay, there might be a point that it will not work for one of the complexes, I'm sure. But for our model, whatever I showed, once we fix the lambda, we have kept it the same for the rest of the DNA protein complexes.
The second question, I'm not sure I completely understand, but it's probably from the paper. How DXY parameter is selected to have an epsilon value for protein?
Oh, I see. So DXY, I see.
Yeah.
I see. So I see. So the DXY actually is an offset. So what it does is when you are computing the free energies of the contact and you're relating it to the contact energies you want all those values to be positive because you are plugging them in the Lennard-Jones potential. So that DXY is an offset that one uses to make sure that all the epsilons that you have are positive.
The last question was, since here you have considered bound state, but can you start it from the unbound states of the
Again, a very good question. In fact, we have done that. So with our model, I mean, it's still going on the simulations. So what we are trying to understand is the assembly process. Like if I have, let's say, the completely dissociated DNA and we have the histone cores, the histone proteins. So the idea is to understand at what sequence you see the different histone cores or the histone proteins being loaded into the DNA construct and how then it sort of wraps around. This is something we are doing, but of course, this is somewhat more difficult than what I have shown you.
The last question from Harsh. So I think also it is kind of similar, but he comments that there are papers suggesting local solvation dynamics around DNA doesn't change much with higher salt concentration, both experimentally and simulation. So the solvation dynamic seems to be independent of salt concentration, but the unwrapping is speeding up at high salt concentration. So yeah, any comment on that?
Again, I think this what you see is because of the disruption of these electrostatic interactions, mostly at high salt concentration. So for example, if you want to do the same thing with divalent ions like magnesium or calcium, you would see the unwrapping at much lower salt concentration as opposed to what you see for monovalent.
I'll just take two more questions, one from Shravanidhi, another Shushmita. Shravanidhi asked, what exactly does the tail do?
Okay, that's again a very good question. So there are various functions of the tails. The first thing, it sort of mediates the interactions between two different nucleosomes. That is one of the things. And it is also basically one of the hotspots for post-translational modifications. So it has a lot of these residues that can be modified in the post-translational sense. So once you do these post-translational modifications like methylation, acetylation, that can have a huge impact on the dynamics of the nucleosome or even the structure or organization of your chromatin fiber, because of these modifications that you do in the tail region, your nucleosome can become more flexible, and then the chromatin then can open up more easily. So this is why the histone tails are extremely important for mediating internucleosomal interactions and for this effect of PTMs. And in the case of the unwrapping, which I showed, where if you chop off the tails, you see a loss in the asymmetry. The reason is this: like when you have the histone tails, and when the unwrapping is happening, after the DNA has sort of unwrapped substantially, then there is then what happens is there is some kind of communication from one end of the DNA to the other end, and then the interactions of that other end of the DNA with the histone tails become much stronger. So, but once you chop off the histone tails, you do not have this gatekeeping activity anymore, because then you have nothing to hold the DNA at the other end. So you see some loss of the asymmetry because there is nothing that prevents the other end of the DNA from unwrapping at that point.
Okay, that's a very interesting aspect. Thank you.
Thank you. So the last question from Shushmita. Shushmita, please go ahead.
Yes. So a very nice talk. We thoroughly enjoyed. Deban, I have questions from a couple of questions actually. So when you are modeling DNA, this is essentially much more analogous to the 3SPN, which is
Right.
modeled
Right.
by long before by Juan Di Pablo. So
Right,
how
right.
is it particularly different?
So yeah, that is a very good question and a valid question. So the DNA model that you have for Di Pablo, it is basically somewhat biased towards double-stranded DNA. So the problem with that model is it does not get the persistence length of single-stranded DNA correct. It gets the persistence length of double-stranded DNA correct, but in their model, the single-stranded DNA is way too stiff. So that is one of the advantages of our model. We get both the persistence length of single-stranded DNA as well as double-stranded DNA correct at all salt concentrations. So, and it has a more, I think our model has a more balanced description of single-stranded DNA and double-stranded DNA as compared to Di Pablo's. But in terms of the resolution, it's the same.
Wonderful. And I could not quite understand that when you said that in your potential you have a native and non-native,
Right.
then what you are doing, you are basically trying to optimize your model according to the B factor. The UNN part I could not quite understand.
Oh, okay. So that part maybe I did not explain well. So the native part, native interaction is just the go part where you see if a particular contact is then. So all other contacts are basically all other interactions we assume to be purely repulsive. So those are the non-native contacts, things which are
so
not.
You are excluded. That is why excluded potential you are assuming, right? That is
yes. That is just the purely
that is
repulsive.
Essentially go like.
Yes. So that the non-native is basically purely repulsive.
Okay, and my final question to you, a quick question, that when you were trying to understand your mutations by, you got arginine and phosphate contacts, which is like the classical interactions that RNA protein or DNA protein makes. But why you are trying to, mutate with lysine? Because it is dipihackyl and it is not region specific. Is that the reason?
No, reason we wanted to do lysine, arginine to lysine, the idea was, we are keeping the electrostatic, the charge is still the same, right? So let's assume.
Like your electrostatic is continuum.
Yeah,
right?
so charge is,
it doesn't account for heterogeneity.
No, yeah, so it's still +1 on lysine. So if I think about the interaction between phosphate and arginine and phosphate and lysine, so lysine also is +1 like arginine. So let's assume if the energy scales of the interactions, like the van der Waals interaction energy scales, if it had been the same, then you would not capture this effect. So we wanted to highlight the sequence specificity of the statistical potential, where this epsilon ij is different for arginine phosphate and lysine phosphate, which is a non-electrostatic component. So we wanted to highlight that, if you don't have the same, if you don't have different.
I wanted to understand if there is any
Susmita, so we are running out of time. Maybe we can continue further discussion later, maybe after the second talk, if possible. So yeah, we will have to now listen to Sayantan. So before that, let's thank once again Debayan. Excellent talk. Excellent. We had very nice discussion. A lot of questions. There are some more questions in the chat box, and I'm going to share those with you later.
Okay.
And I'm sure you can write to Debayan, and you'll be happy to answer your questions. So with that, let's close this this particular talk, and let's move on to the next talk. So to introduce the next speaker, Dr. Sayantan Mondal, I request my co-coordinator, Dr. Rajiv Bishwas from IIT Tirupati. Rajiv, please.
Yes. Am I audible?
Yes. Please go ahead.
Okay, fine. So good afternoon, everyone. So as we are moving to the next talk, next talk is going to be delivered by Dr. Sayantan Mondal. He is excellent budding scientist, and he has done Bachelor of Science from St. Xavier's College, and then followed by Master of Science from Calcutta University. After that, in the year of 2015, he joined Professor Bakchi's lab to do his PhD, and he has done excellent amount of work in protein dissociation, microdroplet chemistry, biomolecular other areas, and did excellent PhD. And in the year of 2021, he has joined Professor Kyung Kui's group in Boston University to do his first postdoctoral research work, where he has worked in mostly coarse-grained modeling of disordered peptides or polyelectrolytes. And after that, he has recently moved to Professor Sakhnovich groups in the Harvard University, and he's working on liquid-liquid phase separations of proteins. So in today's talks, you'll be telling us a story about liquid-liquid phase separation in cells, relevance to intermediate filament formation and membrane remodeling. Sayantan, the stage is yours.
Am I audible?
Yes, you are. Can you make it full screen?
Yeah. Just doing
Yes,
that.
perfect. Please go ahead.
Okay, let's. I think there is a problem. Let me just.
No, it was fine.
No, it was showing the
So-
rainbow
Okay.
inconvenience. Okay, let me share that again.
Okay.
Now it's
Yes.
fine.
Yeah, it's full screen.
Okay. Hi, everyone. And thank you, Rajibda, for the nice introduction. And I would like to thank the SMCB coordinators to give me the opportunity to present my work and talk in this wonderful platform. And keeping in mind the heterogeneity of the audience, so I would like to first introduce the audience to the topic in general, like liquid-liquid phase separation. And then I will spend some time to explain that what is relevance to the intermediate filament formation. And if I get time, I'll move on to membrane remodeling. So liquid-liquid phase separation, by definition, it's a mechanism by which a homogeneous solution of macromolecules, for example, proteins or nucleic acids, they spontaneously demixes into coexisting dense and dilute phases. So the closest analogy to our macroscopic world is the oil in water system, where if you mix oil in water, you can see that they readily phase separate. And if you see microscopically, you can see the oil droplets that are floating inside the sea of water molecules. So this is a typical phase diagram that one can produce. And we generally plot temperature or pH in the y-axis and concentration in the x-axis. And there is this dome-shaped region inside which you get the two-phase region, and outside you get the homogeneous one-phase region. And inside the dome, there is also one region that is called the spinodal region, which is basically the instability that gives rise to the pattern formation, and then it leads to phase separation. And there is this narrow region, which also gives rise to phase separation, but with a different mechanism known as nucleation. So we all know that the organisms, they are made of cells. And if you look into cell, if you take a cross section, and you can see all the organelles and cytoplasm, and the cytoplasm is a very crowded kind of soup of different proteins and other macromolecules. So the organelles, which can be thought of as the organs of a cell, they can be classified into a membrane-bound and a membrane-less organelles. So the membrane-bound organelles, as we have already came across many times in our textbooks, like mitochondria, lysosomes, or endoplasmic reticulum or Golgi apparatus, this kind of organelles, they have a membrane outer layer, and they have a well-defined shape inside the cell. But on the other hand, there are some other organelles, such as processing bodies, Cajal bodies, nucleolus, or stress granules. They do not have any membrane-bound form. So they form and they can dissolve, and they do not have any proper shape. So these kind of things came into highlight like a couple of decades ago. And basically, the membraneless organelles are made out of intrinsically disordered proteins. That's what scientists have discovered. So how do you distinguish between a folded protein and a disordered protein? So basically, you can think of a conformational energy landscape or free energy landscape of a protein. And this is the typical folding funnel kind of picture, which we can think of a folded protein, that there is a global minima, which is deep, and the folded region lies somewhere here. And if you run a simulation or inside a cell, this protein, if it is not drastically changed its condition like temperature or pH, it will fluctuate more or less around this shape. On the other hand, for disordered proteins, you have a rugged energy landscape for the conformational transitions, and the barrier between different wells and valleys, they're actually very low. So that's why rapid transition can happen, and a protein chain can show different conformations. So that's what gives rise to this intrinsic disordered, and basically they lack a three-dimensional structure. So another term that often comes into the phase separation literature is the low complexity domain. So how to understand the low complexity domain? So for example, if you take a well-known sequence, if used in sarcoma, and you just look at the sequence, you will see that there are sequence similarity in different parts. For example, there are 27 repeats of this SYS or GYS or GYG kind of motifs. And if you plot the percentage abundance of amino acids, you can see an abnormally high amount of glycine, and then some serine and glutamine, and some tyrosine. But other amino acids either absent or they are very low in percentage. On the other hand, if you take like a similar amino acid content, like 247 amino acid here, and a carbonic anhydrase, if you plot the same thing, you can see more or less well distributed, and mind the y-axis, they are not the same. So in one line, you can say that the low complexity domains are the regions that shows a composition bias, or it lacks the diversity in different amino acid sequence. And often these low complexity domains, due to their you can think about like when something gives rise to a folded protein, it basically drives the hydrophobic pore formation and lowers the solvation free energy. But the intrinsically disordered protein, they like to remain in this open thread-like state because they are well solvated and they are full of charged residues or polar residues. So that is the molecular reason that some protein sequence become disordered and some become folded. So now if you try to think about the physics of the polymer phase separation, so the very simple level, in a mean field level, you can think about the Flory-Huggins theory. And for a binary mixture, the delta F of mixing, that's the free energy of mixing, is given by this expression, where this summation x ln x kind of term gives you the mixing entropy, and the chi xa xb, where x stands for the mole fraction of each species, and that gives you the enthalpy. So the chi parameter, which is Flory parameter, in turn is related to the different interaction energetics. So if chi is greater than 0 and after a certain critical limit, it basically gives you, so if you plot this delta F mix against phi, which is the volume fraction, and basically you can convert this binary mixture picture into the polymer and solution picture. So you can convert this xa to phi. Phi is the volume fraction, which is related to the mole fraction, and n is the degree of polymerization. So it basically keeps the same architecture of the equation, but some change happens in xa, where xa becomes phi p by n, and n is the like how many monomers are there in a polymer. So basically, if you plot this phi, which is the volume fraction of the polymer, and in the y-axis you plot the delta F of mixing, you can see that with increasing value of chi, it slowly makes a transition from a single well picture to a double well picture. And in certain value of chi, which is the critical value of chi c, it actually makes the transition from a single minima to a double minima. And whenever it goes to a double minima state, it creates two phase. One is rich in say particle type A, another is rich in particle type B, and it starts from a almost miscible state. So this is for n equal to 1, and this is related to the binary mixture case. When n is greater than 1, then the symmetry is lost, but you can still recover the similar physics of creating two different wells starting from one well by increasing the chi. So if you think about the dynamics of phase separation, that how with time, how this progresses. So basically, there is a very nice paper which is published in JCP from Professor Bakchi's group. And they basically did that. They started from a very high value of temperature. And remember the phase diagram where inside the dome, there is the two-phase region. So now they quench the temperature from, say, T star equal to 5 to 0.5 so that they come in the spinodal region. And they saw the three different phases of phase separation. So the first one is the formation of different islands inside the box. And then that is actually the exponential with respect to this kind of order parameter, which is a spatial temporal average of the different mole fraction of A and B in a position R at time t. And so the second stage is the power law behavior, which actually arises from the connection that the islands are making in between themselves. So that gives rise to this kind of patterns that we see as a characteristic of spinodal decomposition. And the last or the third phase is the logarithmic dynamics, which arises due to the coarsening, and that is actually the smoothening of the interface. So it starts from a very rough interface, and then it goes to this kind of smooth interface. So that's kind of the dynamics of phase separation. So this paper is, of course, it's not justified to keep it in just one slide, but due to the interest of time, I am going to move to another point, which is the molecular drivers of liquid-liquid phase separation. Now, when you think about a condensate, which is basically, say, denoted by this kind of spherical dot, and then inside that, you have different interaction. Like the interaction can be charge-charge interaction between charged amino acids like glutamate or lysine or arginine. There could be cation pi interaction, which is a short range, but it's directional. So between, say, arginine and phenylalanine, or arginine and tyrosine. And there could be dipole-dipole interaction, basically between any kind of polar amino acids. And there are pi-pi stacking interaction between the aromatic amino acids like phenylalanine and tyrosine. So all these regions, they can basically give rise to sticky interaction, and they are known as stickers. So this is translated to a very simplified picture of liquid-liquid phase separation that is known as a sticker spacer model. So basically, we have these large beads which acts as stickers. You can imagine that they can be of this kind of amino acid characteristics. And then these cyan beads, they are actually spacers. So they do not have any sticky interaction, but they can involve in the phase separation and increase the liquidity or the fluidity of the condensate. So this is how it works in an animated view. You can look into the video that how the stickers are coming close together, and basically they are forming some long-lived bonds, and the spacers are flexible chains. So that is how this sticker spacer kind of framework can give rise to a liquid-liquid phase separated droplet state. So now I'll highlight some of the open questions and challenges in the field. So the first one is droplet aging or maturation. So what exactly is meant by that? So a droplet aging, by droplet aging, I mean that it changes the rheology of the droplet with time. So basically, it changes its viscoelastic properties. So you can think about a photo bleaching experiment, and this is early stage of the droplet formation. So this is some intrinsically disordered protein that give rise to this biocondensate. And you photo bleach a certain portion of it by using a very high intensity laser. And then you see within like 3 minutes, the region has recovered. That means there is a diffusion inside the droplet, and the fluorophore from other part of the droplet, they actually diffuse and they actually recover that part. But when the same thing is done after 46 hours of droplet formation, so you do the same exercise, you photo bleach a certain region of the droplet, but within same waiting time, like 3 minutes, the region has not recovered back to its original state. So that means the diffusion has slowed down dramatically. And the same thing happens with droplet coalescence. So in the early stage, like within 30 minutes of formation, these two droplets, they can come close and they can coalesce into one big droplet. So that's what you can expect, because they will try to minimize the surface area. But when it is the late stage of the droplet, when the droplet has already aged, then we can see that it's kind of a fused but not fully coalesced droplet. So that means the internal dynamics of the chain has already been slowed down. And this is some quantification of the same thing, that if you track you put a fluorophore tracker, so it explores more phase space, but in the late stage, it just does this much. So there are some explanation that why this can happen, of course, from based on models. And by the way, I forgot to mention that the fate of the aged droplet could be many things. It could be gel, or it could be a fibril, like amyloid fibrils, or fast LC also can form fibrils, or it could be a glassy state as well. So it is not known that how one particular sequence can give rise to so many different kinds of aged droplet, and what is the driving factor for the same. So one simplistic model-based idea you can think about is that if you have a fully mixed state, and this is the early stage of the droplet formation, so now there can be something like sticker saturation. So you can think about Velcros. When one sticker found its partner, then it is not been able to exchange because the interaction is very strong, or the interaction change requires a large energy barrier to, like suppose two phenylalanines are stacked together, so they have to go out of each other and then join with another chain. So if this situation is energetically exhaustive, then we call the situation as sticker saturation. And in that case, there will be no droplet merging, because all the droplets, their valencies are exhausted. And if they are not exhausted, so they are due to Oswald's ripening or coalescence, they can grow into bigger droplets. So, but again, it is not yet known that what kind of sequence can give rise to which kind of phenomena, and what will be the fate of the aged droplet. So that's kind of one open question to the community that people are trying to address. And the second question that I would like to highlight is the polydispersity even before the aging process. So if you think about the oil in water equilibrium, so it doesn't stay like different droplets in the beaker. It always phase separate into one distinct oil phase and one distinct water phase. But on the other hand, if you look into the LLPS droplet, you can see all the experiment, they report this kind of picture. That means there is a polydispersity in the size of the droplet. Could it be a kinetic effect? Because there are some coalescence that are happening locally, but as a whole, they don't form a big chunk of, say, green thing, and they remain as one droplet. So that should be the thermodynamically understandable, because they would like to decrease the surface area. But that's not the case. So, and there is a recent paper by Ned Wingreen's group who showed that the droplet size distribution follows a power law, and I think the exponent was t to the power minus 1.5. X to the power minus 1.5. So basically, so this is a puzzling fact, that why they do not coalesce into one big droplet. So there are some possible reasons. So one is the sticker saturation that is happening before the aging has kicked in. And also, I think I brought up this problem to Professor Bakchi, and he also suggested that this could be another mechanism, that the role of the solvation shell, because these IDPs are highly charged proteins, and when the condensate is formed, there is a distinct solvation shell that will be having a fixed orientation. And so when two of these droplets will come, the solvation shell will repel each other. So that is another mechanism that could play a role in maintaining this polydispersed situation. And this is another like a bigger question, that what role is actually plays in the cellular biochemistry. So we know that what kind of things it can do, like it can concentrate some selected substrate into one place to enhance the rate of the reaction. But again, it is not known that whether it's just confining the diffusion or it's doing something with the activation barrier as well. Or it can create micro compartment inside the cell to act as a micro reaction crucible. And it can control molecular flux inside it to make certain reactions happen, and also hide certain reactant from being exposed. So we know what it can do, but how it is doing it and why there is a need of this kind of membraneless organelles and why it cannot be done in a membrane-bound organelle. So these are the bigger questions that like people are trying to address. So now with kind of a heavy introduction, I would like to come to the work I have been doing at Harvard University in collaboration with David Witt's group at in the physics department, and with Orko, Aniruddha, and Eugene. So basically, we're trying to see that what is the role of liquid-liquid phase separation in the intermediate filament formation. So before going to the intermediate filament formation, we'd like to know that where it actually stays. So basically, the cell have a skeleton, which is known as the cytoskeleton. And the cytoskeleton is a structure that helps the cell to maintain its shape. And it also takes part of when cell divides into two, or the motility of the cell, the profusion of the cell, and all this kind of movements. Cytoskeleton plays a good role. So the cytoskeletal filaments can be divided into three broad classes. The first one is the microfilaments, which is very thin, like 6 to 7 nanometer in diameter, and F-actin falls under that category. And there are the largest kind of cytoskeletal filament, which are microtubules, which can be 23 to 25 nanometer in diameter, and alpha and beta tubulin forms this kind of microtubules. But in between these two range, there is another kind of filament, which previously used to be known as 10 nanometer filaments, because they are 10 nanometer in diameter, like vimentin and keratin and desmins. But now they have a new name called intermediate filaments, because they're intermediate in size between these two. So there is an interesting fact about this intermediate filament protein family, that there are 5 different types of protein. But be it keratin, be it vimentin, be it desmin, there is a common architecture in the protein. So all of them has an N-terminal domain, which is disordered. All of them has a C-terminal domain, which is also disordered. And in between, there is long rod-like structure. And in some places, there are some linkers, which adds the flexibility of the two domains. So this is a representation of different intermediate filaments, which is stained with some fluorescent dye. And the intermediate filaments are shown in red, and the greens are microtubules, and blue are the microfilaments with their actin. So basically, our interest is that, like, what is the speciality about this kind of architecture that gives rise to the intermediate filament structure? So particularly for vimentin assembly, it has been shown that when vimentin filament forms, it basically starts from a dissolved state labeled with the fluorescent dye. And then it goes to some kind of a puncta state, which is basically squiggles. It's just a precursor to the filament state. And after like 3 to 5 days, it goes to the filament state once it is given to express. So basically, our interest is the puncta kind of state, that what is the precursor and why it is like that. And when like liquid phase separation is not that fancy, people just used to call this puncta. But now that we know that a lot of IDRs can form LLPS droplets, so then immediately it comes into our mind that like this puncta state could be some kind of phase separated entities. So there are further evidence in favor of LLPS. So interestingly, there is a mutation in the 117 position. So one tyrosine, which acts as a sticker, basically, if you convert that into a leucine, then there is no filament formation, but the puncta state remains there. Now, if you see this puncta state in this circled region, you can see local coalescence or droplet merging is taking place. And that is a signature of liquid-like property, because if they are not liquid-like, they will not try to coalesce. And that is done by this kind of point mutation. So vimentin exists as a puncta, and we suspect that this could be a LLPS droplet. So recently, actually very recently, like almost a month ago, this paper came out. This is a cryo-EM structure, first of its kind, because there was no cryo-EM or X-ray crystallography data on these intermediate filaments. So they basically showed an AlphaFold prediction works pretty well, because AlphaFold prediction was there before this structure came in. So basically, if you look at a single molecule of vimentin, there is a disordered head region, which is almost like 100 amino acid long. There is a rod region, then there is a linker, there is another rod, and there is a tail region, which has a beta sheet hairpin-like structure at the end. And the total filament, it looks something like this, which is 10 nanometer in diameter. And if you take a cross section of the filament, you can see that the head domains are tucked inside of the cylindrical barrel. And the top view also depicts the same thing, that the disordered head domains, they are actually tucked inside the barrel. So this is a very interesting assembly, and that gives rise to the question that during the formation, what happens first? Like whether the rod domains forms the barrel first and then the head domains tuck in, or it's the other way around, like head domains are making them come together. So those are the questions that were going on for a long time in people's mind. So before going into other modeling studies, if you try to look at just this sequence of the head and the tail separately, and this is the part of the tail which is in the hairpin region. So there are a lot of shading residues. If you take a distribution of the head and tail, you can immediately identify that head has the characteristic of a low complexity domain, because it is rich in arginine, serine, and threonine. And there is like absence of like histidine and lysine. On the other hand, the tail region is rich in leucine, and leucine is known to form like beta sheet kind of structure, polyleucine motifs. So that might explain that why it has this beta sheet propensity, but it cannot be termed as a low complexity region. And there is a server known as this phase separation propensity predictor, which basically, if you give a sequence to the server, the software takes into account all the known IDPs that undergo phase separation, and it matches some part of the sequence and gives you a score, which of course cannot be taken seriously because we eventually have to run simulation and other tests. But it gives you a like preliminary idea that the head domain is drastically higher PSP score compared to the tail domain. That means there are some similarity in the sequence that matches with the previously reported IDRs. So that gives us some kind of a stepping stone. And I am not going to discuss a lot about like why we need coarse graining, because Debayan has already done a nice job in explaining that. So basically, I would like to add one point is that in this project, I use the Martini 3 force field, which is a top-down coarse-grained force field. And by the word top-down, I mean that it is actually a more pragmatic approach and tries to get the experimentally observable quantities right, like solvation free energy and a specific heat like that. And it is a 4:1 mapping. So 4 non-hydrogen atom is clubbed into one big bead. And it has explicit water and ion beads. So like most of the coarse-grained model, they use implicit water and they just mimic the noise of the system as the kick given by the water. But in the Martini model, we have the bead for explicit water, which is like 4 water molecule into one bead. And it has the different bonded and non-bonded interaction. So with that, the first observation is the simulation of the head only and tail only system. And what we observe that the head only system readily phase separates and creates a condensate. And if you plot the gain in pair contacts, like if you take I and j, so summation over all ij pairs, how many contacts are there inside the system. So it starts from 0 and it then gains and becomes stabilized around, say, 400 for the head. But for the tail, it fluctuates very close around 0. So that means the tails are not showing any sign of aggregation, whereas the head only system shows a beautifully formed condensate after 5 microseconds. And of course, this time is coarse-grained time, so not to be tallied with the real time. And this box has water and ion, which will be omitted for the lack of clarity. So then, after doing the head only and tail only system, so we tried to model the whole Vimentin molecule. So it has the head, it has the 2 rod domains and 1 link.
Sayantan,
Yes.
you might have to wrap it up within 5 minutes. Okay, just try to
Okay.
make it. Yeah,
sure. So so it has a head and a 2 rod and linker and a tail domain, and it's an extremely long filament, like almost 40 nanometer length in average. And there is a problem when we throw this in a box randomly. There is a kinetic trap that acts in, because in order to get associated, all the filaments have to be first oriented and then they can come close together. So basically, we took the help of metadynamics to create an initial structure. And due to the interest of time, I'm not going into the details of metadynamics, but it basically accelerates your sampling of the phase space. So once you get the pre-assembled structure from metadynamics by biasing them to come close together, and these greens are the head domains, and this yellow are the linker domains. So basically, I bias the linkers to come together, and the heads automatically come together, but the tails do not. So which also falls in place with the propensity of separation of the heads. But then if you remove all the bias starting from this system, then the heads can remain on their own, and the tails cannot phase separate, or the tails cannot stay in one place, and the linkers also cannot. But there is 2 more interesting facts that do not have any experimental counterpart. So one is that the tails, due to the flexibility of the linker, the tails can actually act with the head. They can get attached, and that can give rise to a multimicellar structure in the bigger framework. And these rod domains, they can almost stay in parallel to each other, which also tells us the propensity for them to form a cylindrical barrel kind of structure. And now the interesting part is that, and this is also experimentally verified, that if you chop off the heads of the Vimentin, there is no filament and there is no puncta even. So we just tried to verify that with our model. And basically, we just deleted the heads manually from here. And then we ran the simulation with the same framework when we saw the Vimentin assembly is falling apart. So that's a 1-to-1 mapping with the experiment. And we wanted to look into the atomistic details. So there is a method by which you can back map the system and basically produce the atomistic resolution coordinates. And then you can see that as there is a lot of serine, so basically I tried to focus on what serine is doing inside the condensate. And what we found is that the serine with itself and the serine with arginine is forming a lot of hydrogen bonds, almost 75 in this case and 100 in this case. So serine and arginine plays a very important role as stickers in this particular system, which holds the condensate together. So what is the test in experimental counterpart? So basically, there is a method called phosphorylation, which is a post-translational modification. And 3 kinds of amino acids in this system are susceptible to phosphorylation: serine, threonine, and tyrosine. And if you phosphorylate them non-specifically, so experiment showed that they do not form the filament and they do not form the puncta or the LLPS state. So basically, we tried to model that, and we added a 1 bead modeled as a phosphate and added -2 charge to it. And then we saw, after some optimization, we saw that it's not forming the condensate when the serines are phosphorylated. So that is also very well matches with the, or complements the experimental result of non-selective phosphorylation. So, and this is kind of an end of the talk. So we're trying to hypothesize that what is the reason for the omnipresence of serine? Because if you look at different sequences, vimentin, desmin, and peripherin, these are 3 same class of proteins, you can see this patchy behavior of serine, like 4 serines together, 3 serines together, and they're occurring either once or twice. And the similar kind of distribution of amino acid, like serine and arginine rich region, is also found in this other type of proteins. And we did an analysis with 254 homologs available for vimentin, and the serine content was average 21%, and arginine is 11%, which is like a very distinct feature of a low complexity region. So this is the summary of this part, which tells you basically the low complexity head domains. They can undergo liquid-liquid phase separation, and which can drive the formation of the intermediate filament precursor state. And serine and arginine can play important role as stickers. And without the head, the vimentin assembly falls apart. And all this conclusion from our modeling is directly verified from the experiment using confocal microscopy. So I had one more part, but due to the interest of time, I'll just skip to the end of the presentation. So I would like to acknowledge this funding and computational resources that we enjoy here, and also the open source softwares that we've used. And with that, I would like to thank the audience for your attention, and I'm happy to take any questions. Thank you.
Thank you, Sayantan, for a wonderful talk. You did an excellent job in introducing the subject. So thanks for that. And I can see a couple of questions popped up in the chat box. So I'll be reading as per their appearance. So first question was asked by Shravanidhi. Shravanidhi asked, how do you define a droplet?
Okay, so the droplet is defined, basically you have to prove its liquid-like character. And one of the proof is that you have to theoretically calculate the radial distribution function, whether it is a solid-like or a liquid-like. You can know by looking at the nature of the radial distribution function. Another thing is for experimentalists, they can do the FRAP experiment, which is fluorescent recovery after photo bleaching. So as I showed, like when like aging, I was discussing aging. So if you photo bleach a region, and as in case of lipids also, you can see that the bleaching region recovers. So basically, the recovery tells you that there is fluid-like motion and diffusion inside the system. So these are some of the ways, and also like the merging of 2 droplets into 1 droplet. So these kind of signatures are used to identify a liquid-like state, and it is not a plaque kind of or like fibrillous state. So
how many protein molecules do you expect to have there?
Right. So this is a very nice,
some approximate number.
Right. So this is a kind of a question that we also wonder, that like how many proteins can make a condensate. So certainly like 2 or 3 proteins cannot do that, or we are not calling them condensates. So basically this is a molecular understanding that we need, and it is lacking in the field, I would say. So basically what we can do is that we can plot a density distribution along the space, and we can see whether we can get a bimodal distribution or a like single peak. So that's kind of the distinction that we can make. But it's a really nice question, and we also wonder among ourselves that like how many proteins it requires to make a condensate or to call it a condensate. So because the cellular limit is very high, so it's in the almost in the order of 100 or 1,000. But in simulations, it's still a debate that which part we call a condensate and which part we cannot.
Thank you.
In a similar line, Rajoshida has a couple of questions. He is asking whether you have seen any coarsening in your simulation or not. If so, then what is the mechanism? And how does the size of the droplet grow with time? Whether does it follow the very famous 1/3 power law behavior or not?
Yeah, so we so basically with a coarse-grained model like Martini, the dynamics is not very trustworthy. But I can show one video which is actually done in the presence of lipid bilayer. But these are the IDPs denoted with red and blue, and you can see and the water molecules are there. So it's basically a ternary system, but you can think that lipid layer is not there because it's almost static and vibrating in one place. So you can see that the little island formation step is there. So where the small clumps are getting generated. And then they are making connections with each other. So which is basically growing phase. And then the rest is done on the top of the membrane surface. So I do not have a quantitative answer because I haven't explored that. But I think the basic features will be preserved as shown in Professor Bakchi's paper. So maybe the time scale will be different, like the ranges where these different stages are seen.
Thank you.
Yeah. So next question is asked by Kankana, and she's asking, from experimental evidence, why you have chosen 117 and why you have modified Y to L, why not other amino acid for formation of.
Yeah, so that is actually not chosen by me or any modeling person. So basically, it is a trial and error method done by experimentalists, and they want to lock the puncta phase. They don't want to see the filament, so they want long-lived droplets. And tyrosine is a unique amino acid that can form two different kind of interaction. One is like hydrogen bonding interaction because tyrosine have OH, and one is the pi-pi stacking interaction. So they work as very good stickers. So it's like kind of a common understanding that if you replace the sticker with something that is not a sticker, and leucine or alanine does the job. So basically, with trial and error, they found out that that kind of point mutation can lock the droplet phase, and it will not progress into the filament state. And there was another question.
Yeah, so there are two more questions in the chat box. I'll finish the chat box, and then Shomak and Shushmita has raised their hand. I will go to them next. So another question in the chat box is asked by Ajit Sharma, and he's asking whether ATP hydrolysis play any role or not in the formation of disassembly or phase separation, whatever you were observing.
Yeah, so ATP actually comes in the phosphorylation phase. So if you see the mechanism, the serine or any amino acid that is susceptible to phosphorylation, they basically get attacked by ATP, and one like phosphoserine is released, and ADP is the other byproduct. So, and this kind of phosphorylation, so what we have done is a non-selective phosphorylation. That means we don't know where the phosphorylation is happening in experimental system. But there is a way to do selective phosphorylation, and cells are actually doing that in order to regulate the wobbliness of the droplet. So basically, for example, if all the serines are together, and if you selectively phosphorylate some of them, so some of the sticker interaction will be modulated, and the liquid nature can change. So that's how maybe the cell is regulating the diffusing in and out of the different substrate by loosening or tightening the droplet with the help of phosphorylation.
So the last question from the chat box is that why you have observed, power law behavior in case of liquid droplets distribution and how it was achieved. That was asked by Ravi Kishore.
Okay, so the power law distribution is actually done in where was the slide? Yeah. So it was done. It was shown in this paper. You can take a look into this paper. So basically, what they have done. So in the like this fluorescent confocal microscopy image, they can assign the sign using some kind of image analysis, and then they can plot the size distribution. And also they have a model by which like they start with different droplet, and by tuning the parameter space, they can get like one droplet phase or multi-droplet phase. And from there, they have tried to explain that which parameter space could give rise to multi-droplet phase. So, but the first inference is from the like this kind of pictures.
Okay, thanks, Sayantan. So I'll move to Shomok. Shomok, please ask your question very briefly.
Yeah, hi, excellent talk. So I have a question regarding the comment you made about the solvation layer or the hydration layer of proteins of IDPs that don't allow them to coalesce. But wouldn't that speak against the proteins forming droplets in the first place? Because, I mean,
yes, but yeah, you are partly right. But the proteins, they do not actually have a shape. So if you think about a single chain, they are like a thread-like and fluctuating. So their solvation shell will not be very static. And also it's not well defined. And the charged patches might not be in one place. But when it forms a droplet, the general tendency is to put the hydrophobic or less hydrophilic residues inside the middle, and then the charged residues on the outer surface. And that's how the charged surface can orient the water molecules, and the water molecules act as a repelling dipole kind of. Or
I mean, if I can just add to that, because I had this question because there is some work by from the group of Parvati Viswas and also from David Leitner, who have shown that the dynamics of the water molecules is actually slower in IDP as compared to structured proteins, right? So I was thinking because it actually forms a pretty strongly bound water molecules in the hydration layer. But yeah, thanks for the answer. Yeah, sure.
Yeah, so it's not a like proven fact. So I'm just guessing
sure.
this could be the reason. So
yeah.
Okay. I'll be happy if it is not as well. So.
Sure.
Okay, thanks, Shomok. So Sushmita, can you please ask your question? Make it brief.
Yes, so Sayantan, very nice talk. I have a quick question that your serine, you left us curious, and I'm curious that whether this whether it is known in the field or not, that whether serine is more prone to become phosphorylated, or is it the more content of serine, is it related to the bending of the filament? Is it known by any chance, or what's the
what so it is known that right. So if you search the whole IDP database, you can find that first is glycine in amount of percentage, and the second is serine. So serine is actually like very abundant in IDPs.
So what is the known characteristics? Serine, is it known by any chance?
Yes. So in the context of vimentin, it is known that if you phosphorylate the serine residues, there is no filament formation. So that is the primary motivation of phosphorylating the serine and not threonine and tyrosine. So there are earlier experimental studies that showed this serine phosphorylation leads to disassembly of the filaments.
Okay. So if there is no other questions, let us thank Sayantan once again for this wonderful talk, and then followed by a very engaging discussion. So now I request my co-coordinator, Dr. Sayantan Chakraborty, to conclude the session.
Okay, thank you, Rajiv, for conducting this session. And so on behalf of all the coordinators, I thank both the speakers, Debayan and Sayantan, for excellent talks and followed by very nice discussions. I'm sure many of you will have many more questions. I can already see that there are some questions in the chat box, particularly for Debayan. So we'll share them with the speakers, and also feel free to contact them. I'm sure they will be happy to answer the questions. And also, I remind you the suggestion from Professor Bagchi that we should have a preprint library, which is an excellent idea. But of course, we need some young volunteers to take up the challenge. So if you are interested to be part of these activities in any way, feel free to contact any of us, the coordinators, and yeah, we'll be happy to take you on board. So yeah, with that, I think we can conclude the session. So as always, today's lecture will be available in our YouTube channel, and the next lecture hopefully will happen within a couple of months. So that will be announced in our mailing list. So if you have not subscribed to our mailing list, please do so again. Okay, with that, thank you all. Thanks for being here till the end. Bye and take
Bye.
care.
Bye.
Yeah. Bye.
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 Bye, sir.